Very good morning to all of you. So welcome once again to some of thoughts and discussion points related to the well-known tumor hallmark, that is invasiveness and metastasis in case of various type of tumor. So today, the whole connecting the dot between whether microbiotas drive metastasis, whether microbiotas distinction at intra-tumoral level or inter-tumor, inter-patient, or even across the species, does it make any alterations in the tumor microenvironment so that invasiveness and metastasis is going to be influenced, and that may lead to the certainly prognosis of cancer patients. So therefore, the interesting point is whether the microbiotas can sense the extracellular matrix in case of tumor tissues, whether the tumor tissue is highly dense, that is the denseness, and the compactness of the tumor tissue. And in an abbreviation, I will use the word DCT, that is the denseness and compactness of tumor. So what? So whether the microbiotas in the tumor tissue is able to sense the nature of ECM, whether the ECM is highly dense, whether the ECM is highly compact, and whether the denseness and compactness of tumor tissues may be associated with the invasiveness and the metastasis at patient level. And that is the whole various questions are there. So today I would like to describe certain mechanism or the proposition, how the DCT, that is denseness and compactness of the tumor tissues, influenced by the nature of microbiotas, may be implicated in metastasis and various other hallmark of the cancer. So we should consider a tumor where the tumor tissue is highly dense. Okay, so here's the point. So if the ECM of the tumor tissue is highly dense and highly compact due to the accumulation of collagen fibers, so then what will happen? And consider here a specific example of the tumor where the highly dense fibers are there or collagen fibers are there. So in other way, the high fibrosis, highly compact collagen fibers. Okay. So what will happen? What may happen? So in the recent propositions are there that high dense collagen or the collagen fibers in the ECM may obstruct the activity of collagenase enzyme, the point number one. The highly dense or compact collagen fibers may also attenuate the DDR1 kinase. It is one of the kinase protein has been discovered to be implicated in fibrosis and the cancer. Then whether the high fibrosis or the highly compact collagen that is linked with the smoking, gutka, environmental factors, that will lead to the unique and distinct tumor microenvironment in the form of DCT, that is dense and compact. tumor tissues. So what will happen? So possibilities are there, I mean, the one way that these dense and compact tumor tissues in the form of high collagen fiber or the fibrosis may have a certain hallmark, that is less metastasis, less invasiveness, and also, in other way, you can say it can be less of, I mean, better outcome in terms of good prognosis. Now, if we look at other way around, other type of the cancer where the extracellular matrix, it shows the accessible extracellular matrix of the tumor tissue where the less compact collagen fibers are there, this way. Okay. The DDR1, as I said, it is one of the enzyme or the kinase involved in the tumor hallmarks. That is active DDR1, means activity of the DDR1 is high. Then various carcinogens, genetic viruses, or mediated transformation may have certain unique type of the tumor microenvironment where the extracellular matrix is highly accessible to microbiotas. So in other way, so the microbiotas, those are pro-metastatic. Those can help cancer cell to go for metastasis. They can go for residing in the tumor microenvironment, and that means that highly accessible extracellular matrix may allow the bacterial cell to go for the attachment, and these bacterial cell that is in the form of unique and distinct microbiotas may help cancer cell to achieve certain further changes or alteration that is in the form of metastasis. In another way, the colonization of intra-tumoral microbiotas within the accessible extracellular matrix of certain tumor type may help towards highly metastatic tumor, correct? No? It can also lead to the high invasiveness, and definitely that will turn into the bad prognosis for the cancer patient. So what we learn in this particular discussion, that across the different type of the tumor, even across the same type of the tumor but different patients, the distinctiveness of the ECM or the tumor microenvironment makes impact. And that is in the form of one of the potential avenues are, that is colonization of intra-tumoral microbiotas. That means certain bacterial cells, those are colonizing the ECM, where the collagen fibers are easily accessible. They will attach to the cancer cells through the expression of some bacterial cell protein, and then it will make, you can say, alterations, and that finally leading to the metastasis and invasiveness. But on the other side, if you look at those tumor or the cancer where the ECM is highly compact, highly dense, like in case of fibrosis-linked cancer, like in case of oral cancer, for example. So in some oral cancer cases where the high fibrosis is there, that may explain that some of the intra-tumoral microbiotas inhabiting the extracellular matrix are not able to work or colonize as efficiently in comparison to the other cancer types where the ECM is not so compact and the dense, and the collagen fibers are highly accessible to the colonizing intra-tumoral microbiotas. So what is the whole question? And there are obvious questions in this particular field is, that is, so whether the intra-tumoral microbiotas sense this denseness or compactness of the tumor tissues? And the answer is yes, definitely, because the intra-tumoral the microbiotas, those are pro-metastatic in nature, are able to sense whether the collagen fibers are highly dense, collagen fibers are accessible, so that bacterial cell can colonize efficiently, and that can lead to the metastasis. So the question is here that microbiotas residing in the micro niche of oral cancer differ from the microbiotas, those in the fibrosis-linked oral cancer. That's what I said. So the heterogeneity within the oral cancer from one type of the oral cancer where it is highly invasive and metastatic, and other oral cancer types where it is less metastatic can be explained by the intra-tumoral colonization by microbiotas. That means the colonies of the microbiotas are different from oral cancer. Those are highly metastatic, caused by mutations, caused by, you can say, carcinogens, caused by tobacco. Other side, those oral cancer caused by fibrosis mechanism, like through the gutka or the betel quid, right? So that can explain the heterogeneity within the oral cancer types. The second is that less dense collagen fibers, whether invite or promote the colonization of certain pro-metastatic microbiotas. That is the question number two. Then whether the highly compact fiber in the fibrosis-linked oral cancer may prevent to an extent the colonization of pro-metastatic intra-tumoral bacterial cell or the microbiotas. So there are many questions still has to be resolved, but one thing is very clear, that colonization of intra-tumoral microbiotas, not only for oral cancer, even any cancer types, may impact the degree of metastasis and invasiveness. And recently, for one paper published in Nature that also elaborated on the importance of intra-tumoral microbiotas that can impact the heterogeneity in cancer. Specifically, it can be oral cancer or any other cancer types. So the whole thought is whether metastasis driven by microbiotas is true, and does it make any sense? And it appears to make a sense because the heterogeneity within the colonies, and you can say the types of microbiotas, is also very, very important between one cancer type to the other cancer types, between one patient to the other cancer patients. And most importantly, it is important to be discussed here, the microbiota's heterogeneity across the cancer patient can also be linked with these questions. And the question is here, whether the tumor microbiota heterogeneity at interpatient levels may influence the prognosis of cancer patient. It may be true, but yes, investigations are needed. The second question is whether the intra-tumoral, right? Inter-tumoral, and most importantly also to understand here, xeno-tumoral microbiota heterogeneity are contributors towards susceptibility, resistance to the drugs, prognosis, and many more tumor hallmarks. And in near future, that can be one of the interesting area of research where at the junction of microbiota heterogeneity, tumor heterogeneity, cancer cell heterogeneity should be discovered and well connected so that it can be useful for the patient for better prognosis. Thank you very much for listening. Keep growing and enhance your thought process in the field of science and research. Thank you very much.